Vaccines. It seems like with every day, there seem to be more and more crazy conspiracy theories out there on the internet about them. You have people on one side telling you they're great and there's no issues, and you have people on the other side telling you they're terrible, and they have microchips from Bill Gates, which you know deep down is completely ridiculous. But you ask yourself, how do vaccines actually work? And what is the science behind them? Well, let's talk about the science behind that.
Welcome to The Science Behind That with Atticus Hamilton.
Hello, all you scientists. Welcome on back to The Science Behind That podcast. I am your host, Atticus Hamilton, and I hope all of you guys are doing well. As you heard in the intro, on today's episode, we are talking about vaccines. We're going to be talking today about the 6 different types of vaccines, what they are, how they work, why they are one of the greatest feats of modern medicine in human history. We're going to be talking about all of that stuff. We're also going to be debunking some conspiracy theories about them. So, ladies and gentlemen, please go grab yourselves a nice steaming hot cup of coffee, add a little bit of maple syrup to it, and we will jump right in. So vaccines. So there are 6 different types of vaccines, and before, I jump into this too much, I just want to say there are going to be some things in this episode that I can't say. Luckily, my last episode did not get pulled down by YouTube, but as many of you already know, the episode before that did get taken down by YouTube, so YouTube is trying to censor my content here, which is really unfortunate because all I strive to do in each episode is just to teach everybody that listens to it something new. But because of that, ladies and gentlemen, I'm gonna have to be cautious about what vocabulary choice I use. So, if any of you are like, oh man, this feels weird, Atticus is, he's parsing his words a lot, that's why. It's because of YouTube. Additionally, for my YouTube audience and everybody else for that matter, I know that a lot of you that listen to my podcast on YouTube, listen to it there because it's more convenient. And the whole reason I put my podcast on YouTube in the first place was to make sure that, people that didn't have access to a paid streaming service like Apple or Amazon or Spotify could also listen to my podcast. However, my podcast is also up on iHeartRadio. So for my YouTube audience, if you want to be sure that you're not missing an episode, definitely check out either my website, which is in the description below, or go to iHeartRadio. It's totally free there, and you can listen to every episode, or you should be able to listen to every episode there if you're worried about missing my episodes. And of course, as we talk more about these scientific concepts that I guess are now politicized or are becoming politicized, there's definitely going to be a greater chance that my content is going to get censored more. So just as a heads up, you can always go to my website or listen on Spotify or Apple or Amazon or iHeartRadio where I'm not censored. That being said, ladies and gentlemen, let's jump right in here. Sorry, I had to take a sip of my coffee. So there are 6 different types of vaccines. There are inactivated vaccines, live attenuated vaccines, mRNA vaccines, which is what the COVID vaccine was, subunit vaccines, and those also include recombinant polysaccharide and conjugate vaccines, toxoid vaccines, and then viral vector vaccines. And we're going to be talking about all of these today. And the genesis of this episode was because Robert F. Kennedy Jr. said that there are no safe and effective vaccines, according to him, which is complete nonsense. There are many safe and effective vaccines out there, and probably one of the greatest success stories of vaccines is the smallpox vaccine. That is probably one of the greatest success stories. And as we jump in here, I also wanted to say this for the YouTube algorithm as well. YouTube, why am I talking about this? I'm talking about this because people need to know that vaccines are safe and that all the conspiracy theories behind them are nonsense. And again, for YouTube, I have years of working experience in the healthcare industry in terms of clinical bacteriology, virology, and parasitology. I've worked in the federal government sector in the areas of national security and biodefense. I've worked in the state sector in the area of public health, and I have my degree in medical microbiology. So there you go, YouTube. All right, so jumping in here, we're going to start first with inactivated vaccines, and I'll give you some examples. So some examples of inactivated vaccines are like flu and polio. Now, how does this work? And I do want to say as well, flu vaccines once upon a time were live attenuated. It's just these days they are inactivated. So basically, all an inactivated vaccine is it is a vaccine that uses the killed pathogen, the killed infectious agent that you're talking about, and you just inject that into your muscle tissue. Generally, there are some vaccines that go up your nose or in your stomach or elsewhere, and your immune system will recognize that dead agent and begin making antibodies towards it. Inactivated vaccines are very, very safe, and they are very safe because the pathogen in them is dead. So there's pretty much no chance that they will cause infection. But they are very safe. The trade-off with this, of course, though, is that inactivated vaccines are not as effective as live attenuated vaccines. Now, what do I mean by this? What I mean by this is, when we look at effectiveness in vaccines, we look at effectiveness in two different ways. Number one is, does it prevent communicating the disease? And number two is, if it does not reduce, or if it does not prevent communicating the disease, does it reduce the severity of infection? And that's how we look at effectiveness. And what causes that is generally the strength of response the immune system gives to that vaccine. And in general, inactivated vaccines induce less of a significant immune response than do live vaccines. And there are a variety of reasons for that, that go back down to pathogen host dynamics and pathology, that are just out of the scope of this episode. You can literally, and I have taken entire classes on this topic. But overall, it's just important to note that inactivated vaccines, while they have less risks associated with live attenuated vaccines, they are, as a result of just their mechanics, slightly less effective than live attenuated vaccines. However, overall, they are still very effective. And so some examples of inactivated vaccines would be like your Hep A, your flu shot, your polio, and your rabies. Rabies especially is inactivated because rabies, there's just way too much risk to develop an attenuated vaccine of rabies, that it's better off to just have the inactivated form. Moving on to live attenuated vaccines. Live attenuated are basically live weakened vaccines. So what they do is they use the pathogen that's causing the illness you want to prevent or you want to vaccinate against, and it's weakened to where it is unable to cause disease in humans and it's unable to spread from one person to another. Now, how does this happen? There's a variety of ways to attenuate a pathogen. There's a variety of ways to do it. So, one way that you can do it, and I'll use this as an example of flu, is with eggs. So once upon a time when flu vaccines were live attenuated vaccines, the way you would produce those is basically, and this is a very simplified explanation, but you would take your flu virus and you would infect a chicken egg or a chicken embryo. Um, you'd let the virus grow in that chicken embryo, and then after a certain amount of time, you would take that embryo, pulverize it up, filter it out, and that supernatant or whatever, or that fluid that went through the filter, that only contains the virus, but it only contains virus that was able to infect the chicken embryos, which means now they're better equipped to infect chicken embryos than they are human cells. And then you take that solution from egg 1 and you inject it into egg 2. Then you take egg 2 solution and go to egg 3. So, and you do this over and over and over and over and over again. So you're essentially creating a serial dilution here. And at the end of that cycle, what you end up having is you have a live virus, but because you've passed it so many times through so many generations of chicken embryos and of viral generations, you end up having a virus that's unable to infect human cells because it has essentially evolved in your lab to infect chicken embryos. And that is the most basic way to attenuate a virus and to create a vaccine. Now, there are a lot of other ways to do this that I'm not going to get into, but that's just a basic idea of how and what attenuation is. So all it is it's a weakening of the pathogen to make it less pathogenic and less infectious. And, when we look at live attenuated vaccines, they are generally more effective than inactivated ones because they elicit a stronger immune response. However, they need to constantly be kept cold. So when we look at vaccination efforts in third world countries, they're a lot less practical because you have to keep them cool. So, if you're going to an area with limited refrigeration, you need to be able to store them in portable refrigerators. And of course, some individuals may experience adverse side effects, such as people that are immunocompromised. And that could be for a variety of reasons, but people that are immunocompromised with live attenuated vaccines, there is a chance, albeit a small chance, but there is a chance that pathogen could revert back to wild type and infect that person. But a very low chance. And then some examples of your live attenuated vaccines, your MMR vaccine, rotavirus, smallpox was a live attenuated vaccine, chickenpox, yellow fever, things like that. Those are your live attenuated vaccines. These are diseases that you really need a good, strong immune response to create a very effective immunization agent. So now let's talk about messenger RNA. And I know that there were a lot of conspiracy theories back during the big Rona about mRNA vaccines. I did an entire episode just about the COVID vaccine a long time ago. So if you're interested, definitely go back through my history and listen to that episode. That was a really good episode. But in a basic sense, messenger RNA vaccines or mRNA vaccines, all they are is basically a little bubble that contains a strand of mRNA. And this mRNA encodes the spike proteins, or the outside proteins on the surface of the virus, that attach to the cell receptor that grants entry of that then results in the cell granting the virus entry into the cell. So all the mRNA is it just encodes that one protein, or those one proteins that are the spike proteins for the virus. And so what happens then? So when you get immunized with the COVID vaccine in this situation, or a messenger RNA vaccine, that messenger RNA goes into your cells, and your cells read that and translate the mRNA into protein. And those proteins are the spike proteins for the COVID virus. And then those cells display those proteins on their outer cell membrane surface. And then that's when the immune system sees it. They bind to those proteins and create the immune response. And again, I did a whole episode on the fundamentals of vaccine and immunity development. But that's how they work. So I know that there's a lot of conspiracy theories out there, like they change your DNA and all that stuff. Utter nonsense. Do not listen to any of that. Absolute nonsense. Human cells do not go from mRNA to DNA. They don't. Now, there are some examples of that. It's called reverse transcription. There are some examples in other organisms of reverse transcription. One such example being retroviruses. Retroviruses, their genomes are in RNA, and they go from RNA to DNA. But human cells do not. And in the COVID vaccine in particular, this mRNA that is in there is literally just encodes for one protein, which is the spike protein on the surface of the virus. So it's safe, it's effective. However, there's something to note with the COVID vaccine, which is kind of interesting, because the COVID vaccine does not necessarily reduce the risk of getting significant. It does not prevent you from getting COVID. And that was an interesting thing that came out after the development, was that unlike other vaccines, which significantly reduce the likelihood that you even get the pathogen in the first place, the COVID vaccine doesn't really do that. It does reduce the likelihood that you get the pathogen, but it's not by 100%. And the primary function of the COVID vaccine is to actually reduce severity of symptoms. So instead of being hospitalized for not being able to breathe, it's more like a little cold. And that's what the vaccine does primarily, is it reduces the severity of the virus when and if you get it. And so far, the best example of a messenger RNA vaccine is COVID, which is kind of cool. I mean, the actual science behind it works is really innovative, that they were able to identify exactly what mRNA sequence creates that one protein, that one spike protein of the virus, and that they're able to package that in a liposome and deliver that in vaccine format is pretty cool. So, next, we're moving on to subunit vaccines, recombinant vaccines, polysaccharide, and conjugate. So all these they are pieces of the pathogen, much in the same way that the messenger RNA vaccine produces just one spike protein. The subunit vaccine is that protein, or it would be that protein. But it could be proteins on the surface of the pathogen. It could be sugars. It could be a capsid that circles the pathogen. It could be any variety of things, but a subunit vaccine is just bits and pieces of these pathogens that are then mixed with adjuvants, which are compounds that are explicitly designed to elicit a strong immune response, that are then injected and the immune system recognizes them. So then if that person becomes exposed to that pathogens, for example, pneumococcal pneumoniae, uh, then the immune system is able to recognize the subunits in that vaccine against that agent. So because of this simplicity, if you will, it elicits a very, very strong immune response to only one part of the pathogen, which if you're talking about DNA viruses, which are pretty stable, or bacteria, or, yeast or something, it's good because they don't really mutate very much. And the other benefit of this is that there's no chance of it causing infection or anything like that. The problem is that with a lot of these subunit vaccines, you need booster shots. You need booster shots because the antigen profile on the surface of these pathogens changes over time. And unfortunately, the memory of these pathogens for subunit vaccines isn't as long as something like a live attenuated vaccine memory would be. That being said, it is very effective against things like HPV, Hep B, Haemophilus influenzae type B, whooping cough, pneumococcal disease, meningococcal disease, shingles. These are all subunit vaccines that are very effective. They're very safe and effective. It's just they have, like everything, there's a cost-benefit analysis here where, they are more effective than inactivated vaccines in some ways and less effective in others. Second to last one we have here are toxoid vaccines. So toxoid vaccines are vaccines against a toxinous product that a pathogen produces. So some pathogens out there, they cause disease not through eating away tissue or infecting cells, but they cause disease through the production of a toxin. Two examples of this are diphtheria and tetanus. And these are classic examples. Another one would be botulism, right? Botulism, Clostridium botulinum produces the botulism toxin. And when somebody has or is infected with Clostridium botulinum, it is the production of that toxin, the botulinum toxin, that causes their symptoms. With toxoid vaccines, the vaccine is using either the full toxin that is bound to another agent to neutralize it, or it's using a subunit of that toxin that then functions much like a subunit vaccine does in creating immunity to parts of that toxin itself. So then when the person is exposed to tetanus, for example, their immune system produces antibodies to neutralize the tetanus toxin, which is Clostridium tetanii. So again, with these vaccines, you need to get booster shots. And again, two examples would be your diphtheria and your tetanus. So viral vector vaccines are the final one that we're talking about today. They are pretty new. So what they are is they're vaccines for things like Zika and flu and HIV that use a modified version of a different virus to deliver generally like mRNA to a cell, and then it would deliver that mRNA to a cell, and the cell would produce the mRNA and express the right protein on the surface of the thing that you're immunizing against. And so for these viral vector vaccines, a lot of different viruses have been used as vectors, including, VSV, influenza, measles, adenoviruses, and they are being experimented with in the development of new COVID vaccines right now using adenovirus as a viral vector. With these, there's, again, very little chance of developing illness because you're not actually infecting a person with the virus you want to immunize them against. You're infecting them with a genetically modified different virus that's only there to deliver a short sequence of mRNA, which can then be used by the cell to produce a protein that's going to be the focus of the immune system in developing the immune response. So, ladies and gentlemen, those are the types of vaccines and how they work. So there's 6 types of vaccines, as we discussed, and we also talked about how they work. Now, really quickly, I want to address some conspiracy theories out there. There are people that believe that certain mental health issues are attributed to vaccines. They are not. Those ideas came from a doctor way back in like the 60s in the United Kingdom that published a bogus report linking the two together, even though it was complete nonsense. And that physician was discredited. He lost his medical license because it was demonstrated that he made it all up. He made it all up. Vaccines also do not do any of the other things that JFK said, JFK Jr. said they did, or said they do. He has said a lot of conspiracy theories about vaccines, and they do not do that. They don't do any of it. Vaccines are one of the most effective and innovative public health measures and pieces of medical technology that we have ever created. The first vaccine was created by Edward Jenner, and it was basically cowpox, and it was used to immunize people against smallpox, which I'd like to remind everybody, in the 20th century alone, smallpox killed over 500 million people. But because of the development of the smallpox vaccine, that was one of the 2 diseases ever to be eradicated by humanity. And the last natural case of smallpox was in 1977 because of vaccines. Vaccines saved countless lives in their development. So, ladies and gentlemen, of course, as with everything, there is a lot of misinformation out there. Unfortunately, I really wish there wasn't. But there is a lot of medical misinformation out there. And at the end of the day, the reality is vaccines are safe and effective. And yes, they have side effects, but so does everything. Every medical thing, everything in existence has side effects. And to be honest, because of how much scrutiny people have given vaccines for some inexplicable reason, they are probably one of the safest medical interventions out there. So, ladies and gentlemen, with that being said, I really hope you enjoyed today's episode. I had a lot of fun making it. It's kind of sad that I have to make this episode. It's sad that there are that many people and people of power that believe in these conspiracy theories, that they do this and that and the other thing when they don't. But ladies and gentlemen, I hope you enjoyed today's episode. I hope all of you have a fantastic weekend. Get some rest, relax, chill a little bit. We all need it. And I will see you all on Monday. And remember, as always, ladies and gentlemen, stand up and question everything.